So that's what we have in mind for today, and we'll move right into the meeting. And we couldn't be starting this meeting off any better than with the talk that we're going to hear, because this talk is about sleep. So for those of you that are not fully awake yet, you better wake up. So Chiara Cirelli is a faculty member in our department, and I'm very proud of having her as a faculty member. She's just been absolutely outstanding in our department, and has recently taken over as vice chair of research in the department. We were really very fortunate to get Chiara from the Neuroscience Institute out in San Diego a number of years ago. I think it was around 2001 that she and also Giulio Tononi both came together to really form the basis of what now I think is an internationally known group related to sleep and consciousness that both Chiara and Giulio have been very involved with. Chiara's work has been on the basic side and the translational side, as you'll hear, asking very basic and fundamental questions about what are the mechanisms by which we sleep, what are the effects of sleep deprivation, something of which at least I can tell you that I've been experiencing a little bit recently. And how does this all work together? What are the molecular underpinnings of that? And very importantly, what is sleep for? And what you'll hear from Chiara is what now is not a novel hypothesis, but many years ago was a very novel hypothesis about why we sleep and what the molecular changes are during sleep that help us do better during the day, and what happens if we don't get that kind of sleep. So Chiara graduated from the University of Pisa with an MD and a PhD. And did a postdoc there related to, I was reading it in Italian, so I couldn't quite get it, but I think it was related to the noradrenergic system and REM sleep and wakefulness. And then, as I mentioned, moved to San Diego and was a fellow at the Neuroscience Institute there, and then joined our faculty in, I think, 2001 or 2002. And as I mentioned, it's just been terrific to have Chiara on our faculty. She mentors many graduate students, is on many committees, and is one of the most supportive scientists in our department, and that our students like to go to. She came here, quickly moved up through the ranks, has done all kinds of interesting work. I'm very into animal models, but I never realized that we would have actually flies in our department. So when Chiara came, she set up a Drosophila laboratory right next to our clinics. And every once in a while, we'd see a little fly emerging somewhere, and we'd always say, "Where's Chiara?" But as you'll hear, she's worked with a variety of different species, including working with humans to some extent. Most importantly, in relation to her international recognition, she recently received a number of awards, including an award from the Pisa Sleep Award, which is a very important award, in 2022 for outstanding contributions. And she's going to be receiving in June the Distinguished Scientist Award from the Sleep Society. So very much congratulations, Chiara. So without me going on any further, let's give Chiara a warm welcome and listen to what she's got to say.
Good morning, everybody. I see you are all awake, and I hope you will stay that way. It's a great opportunity for me to be here today. Thank you very much, Ned and Richie, for this opportunity to tell you a little bit about our efforts to try to understand why we all sleep. And first of all, first I want to acknowledge, as Ned alluded to, that all the work that I'm going to present is done in collaboration with my long-term collaborator, Giulio Tononi, now at the Wisconsin Institute for Sleep and Consciousness at the Department of Psychiatry here in Madison. And all the work is funded by NIH and the Department of Defense, and I have no conflict of interest to declare. So I want first very briefly to remind everybody why we in the sleep field think the sleep is very important, and a little bit about some ideas why we think so and what the sleep is for. And then, of course, I will focus mainly on these hypotheses that we have been testing about synaptic homeostasis. So why do we believe sleep is important? Well, one, first piece of evidence that a strong, very strong evidence comes from phylogeny. Every animal species that has been carefully studied so far sleeps. And so I'm not only talking, of course, about mammals in general and birds, but, you know, bees, flies, of course, sea snails. And you can see perhaps on the top corners, recently, the flatworm or the jellyfish Cassiopeia have been shown to meet all the criteria that we are using in mammals to define sleep. The second very important fact is that we know in the brain there are very complicated mechanisms in place for the homeostatic regulation of sleep, meaning that there are systems in the brain assuring that if we lose sleep, if we're sleep deprived, we then have the ability to recover at least some of the sleep that was lost. And this is not just, or usually, at least in mammals, it's not so much in terms of sleeping longer, but sleeping deeper. So although there are still maybe a few people who believe in the null hypothesis that sleep is just a time filler, I think this evidence strongly argues against that, because why would you have a homeostatic complex system to recover sleep if sleep were just a time filler? What is sleep for? You can say, well, we all know, and Ned perhaps alluded to that. If we don't sleep, the next day we feel really miserable. Everything is difficult. The mood is down. In fact, there are a lot of studies, especially in humans, where this kind of analysis is, of course, easier to do, showing that it is quite hard to actually identify a single aspect of cognition that is not affected by sleep deprivation. So it's attention, is the ability to learn and to consolidate memory, but is also the ability to integrate a lot of different information, is humor appreciation, mood in general, risk control and assessment. And recently, that's a kind of a new area of research, is also becoming clear that even when you test a subject and actually the performance in specific tasks is maintained after sleep deprivation, the subject will tell you that it took so much more effort to get there. So the question of effort allocation after sleep loss is quite a new area of research in our field. Another very important piece of evidence, I won't go into the details of these studies, but it's just to tell you that now we know also that once, because you have been awake too long, or you had, so to speak, a very intense wake period with a lot of learning, and therefore the performance is impaired, you can recover from that impairment only by sleeping. A period of quiet wake, even with eyes closed, is not enough for the recovery. And these two studies, I'm quoting them simply because they have used very different tasks, a visual task, another is a rotation adaptation task, but the result is exactly the same. A nap can do it, 1 hour with eyes closed cannot. So we are back then. What is exactly in the brain that needs to recover, and why can this be done in sleep but not in quiet wake? And we have always thought that if you want to address this question, you need to start from the fundamental feature of sleep, the one that really distinguishes sleep from any other behavioral state, including quiet wake. And that feature is, as shown here in this painting by Rousseau, partial sensory motor disconnection. By definition, when we are asleep, we lose the ability to promptly respond to potentially a very dangerous situation. So you can have any idea you want about sleep, but that idea has to explain why, through evolution, we would have maintained sleep if the same function can be performed during wake or during quiet wake. Because in quiet wake, when you are still able to respond, of course, you are much less at risk. And so we are suggesting that that fundamental function that can only or much better be done in sleep than in wake is maintaining an overall balance of synaptic weights across the brain. So to do this process that we call of synaptic renormalization. And when I say that, most of what I'm going to tell you about is about the mammalian brain, but as you will see, I'm going to discuss also a little bit about the fly work. In a mammalian brain, the great majority of synapses are glutamatergic excitatory synapses. So they look like what you see here. They are axospinous synapses. So there is the presynapse, which is the axonal button, and the post-synapse, which is the spine head receiving the and responding to glutamate. And you see on one side, just one slide taken in our lab using serial electron microscopy of one of these axospinous synapses. So there are many of them. Here you see the calculation that has been done for the human neocortex alone is 100 trillion of these synapses. And we know that in the brain, the bulk of the energy in the brain goes to sustain synaptic activity. There are different estimates because it's actually quite difficult to really figure out exactly the cost, but all estimates agree that the majority of the energy in the brain is used to sustain synaptic activity. So this is something that has to be under very tight control because we also know that the brain uses overall a lot of the energy relative to the body. So this is the general idea of this hypothesis, that when we are awake, for us humans, during the 16 hours or so of our wake day, we are always learning something. It's not just when, you know, we go to a lecture and hopefully today you remember what I'm going to tell you. But in general, by definition, when we are awake, there is always ongoing what is called also incidental learning, because by definition, you are exposed to an environment that is always changing and you need to adapt to that. And that is plasticity and learning. The hypothesis says that because of this process, by the end of the wake day, many circuits in our brain get strengthened. So the overall efficacy of the synapses on average increases. And that's what you see here in these red lines in many brain circuits. The job of sleep then would be to bring back overall synaptic efficacy to a lower level that in the long run is sustainable. For the reason that I just mentioned, you know, learning is good, learning requires overall potentiation, but you can't just keep going up and up because it's too costly. And so this process of renormalization, we think, happens during sleep and is, as I will tell you at the end of my talk, not an aspecific process. We call it a smart down selection because the brain has to be able to select what to weaken and what to maintain at a certain high level, of course, to maintain the learning and then to produce the memory consolidation. It sounds like kind of magic, but actually is not. And we are starting to understand at the molecular level how this could happen. So these are the 5 major points, the rationale underlying this hypothesis. As I mentioned before, we need to learn when we are awake, because it's when we're awake that we need to adapt to the environment. And most not all, but the great majority of learning happens through synaptic potentiation by strengthening synapses, not by weakening them, which brings about, of course, a need for this process of renormalization. Now, these 2 points are not controversial. In the learning and memory field, even in the computer network field, everybody agrees. Learning is mainly through strengthening, and you better implement also rules of renormalization to correct for the consequences of uncontrolled learning. But we are saying the process of renormalization specifically happens during sleep, and it is in fact because of this process that we can account for all the beneficial effects of sleep at the cellular level as well as at the system level. So here is just to remind you, I won't go into the details, but when you study specifically tasks, usually in animal models, and you can study the synapses, and you find indeed that most forms of learning happen through synaptic strengthening. Second point, it's very obvious there are dozens of studies showing that if you implement these rules in a computer network, in a computer, immediately you need to do also the renormalization, so implement rules of weakening or depotentiation. If you don't, like you see here, synaptic potentiation will get to the top. There will be runaway, so-called runaway potentiation. All neurons will start responding to any input. There will be saturation, hypersynchronous, epileptic type of activity. So this is very well known, but many people still think that the brain, or a computer for that matter, can handle this problem by simply implementing weakening of some parts of the circuit or some synapses online while you are strengthening other parts of the circuit. So let's say in the limit now, you are strengthening some connections to learn what I'm telling you. At the same time, you are forgetting other stuff to maintain the balance. While we are saying this is not how it should happen. There is a time, so the balance is there, but is not every second, every minute, or every hour, is across the 24-hour cycle. So there is a time, the wake day, in which overall there is a net increase in synaptic strength, because I'm here in a new environment. Today I took a walk, I thought about what I should tell you today, I thought about the invitation by Ned and Richie. All these are memory traces that they are laid down that I will still remember tonight. The fact that when I'm awake, all the neuromodulatory systems, the arousal system, the noradrenergic, dopaminergic, histaminergic, orexinergic system are all much more active than during sleep. This biases the brain towards learning and potentiation. That's all good. Then sleep happens. And we are back to this fundamental feature. When we are asleep, we are disconnected from the external world. So suddenly we are no longer slave of the here and now. The neuromodulatory systems, here is, I'm just using paradigmatically the example of the locus coeruleus, basically most of them shut off. So the fact that they are not active instead biases the brain towards depotentiation. But very crucially, and I think this is perhaps the single most important finding in our field, in the sleep field over the last century, the brain is not silent. Neurons are actually active, just a little bit less than in wake, but they are active. And for most of the brain, they are all active at the same time. They are synchronously firing or not firing in a very specific pattern, which creates the best condition to actually sample in a very comprehensive way all the synapses in the circuit because everybody is active. And we think that's the moment in which then you can do this smart down selection. Again, it sounds a little bit a miracle, but I'll tell you what we know of synaptic tagging in a negative and a positive way that actually can do this selective process. So everything is based, though, on the first claim of this hypothesis that, well, overall, there should be a net increase in synaptic strength after wake, and then a net decrease after sleep. If we cannot find and prove that, then the entire hypothesis is wrong to begin with. And so we have spent a lot of time over the last literally 20 years trying to measure synaptic strength in the brain and see how it changes at the end of the major wake period and then at the end of the major sleep period. You can ask why it took so long and we are still doing it. Well, it's because unfortunately, there is no single measure of synaptic strength that you can use that everybody accepts and one is enough. There are several measures, some are better than others. Our approach has been to try to use them all or most of them and see whether at the end the evidence is consistent. So I'm going to tell you about electrophysiology, molecular markers, or ultrastructural markers. Also, as I told you at the beginning, we know all animals sleep. And so if this maintaining synaptic homeostasis is a fundamental function of sleep, it should apply to flies as well as to rodents and humans. And that's what we are trying to test. So I'm give you some examples of the evidence that we have been collecting, starting from electrophysiological evidence. So classically, if you stimulate neuron A and then you record from neuron B, the first monosynaptic response that you record, that you see here, the amplitude for a fixed stimulus, a fixed intensity, the amplitude of that response or the slope of the response, as you see here, reflects synaptic efficacy. So, you know, you can do this. We have done this in rats by applying electrical stimuli to their brain, to their cortex, or you can actually do, as shown here, through TMS, non-invasively in humans, applying a small pulse to the surface of the scalp and then recording this monosynaptic response and the slope of the response. And what you see is this change in the slope of the response that goes up with time spent awake. The red or pink column that you see is a night of sleep deprivation. So it goes even higher after a night being awake. And then it goes down when the subjects are finally allowed to sleep. So this is evidence that is suggestive, is consistent with this hypothesis. I'm showing this first because this is the weakest evidence, because there could be other explanation for this result due to the balance between excitation and inhibition, or due to changes in intrinsic excitability of, for instance, leak potassium currents. You could get the same result. There is a more direct way of testing this electrophysiologically, which is by looking at the minis, the miniature excitatory postsynaptic potentials. If you in a slice record without any stimulation, you just record activity, you have these little events, that's why they're called minis. The frequency and the amplitude of these minis reflects both pre and postsynaptic strength, because it depends on the ability of the neurons to spontaneously release, in this case, glutamate. And so this is actually quite an accepted marker of strength in vitro. And what we did in this original study was to take the brain of rats or mice after 6-7 hours of wake, spontaneous wake. At night, rodents are nocturnal, so they are awake at night. or 6-7 hours during the day after sleep, and just look at these events. And I think it's pretty obvious that, you know, both the frequency and the amplitude of these events is higher after wake. Now, I won't go into the detail, but this is just to say that, you know, I think any finding is not really true until it gets replicated. And so these are 3 different experiments by 3 independent labs, basically testing in other areas and finding the same overall result, that the frequency, especially or the amplitude of these events, is higher after wake relative to sleep. Now, what about molecular markers? Those are very accepted markers of synaptic strength. Depending on the species, you can use mainly pre-synaptic markers or post-synaptic markers. The overall rationale being, if synapses are stronger, they have more of these synaptic proteins that are very important for the job done by the synapses. So in flies, we used a marker which is called BRP, Brook Pilot, because it is the best established. It's basically expressed in all Drosophila synapses. And what you see on the left is how we measure the overall levels of expression of this marker in a control fly, meaning a fly that slept the entire night. Flies are like us, they are diurnal, or a sleep-deprived fly that was kept awake the entire night. And you see overall the pink reddish means higher expression. There is much higher expression of this marker after wake than after sleep. The study on the right, that's a different study in which then we focus, because on the left you see is basically what you see is a confocal image basically of the entire fly brain. And so this increase in synaptic strength is quite widespread. Then we focus on a very specific circuit, these VS neurons. They are visual neurons, very well studied in the fly. And basically what we found is that in these neurons, both the number of the spines, which means the synapses, and actually the branching of their dendrites increases when flies are awake, and especially, and that's the first EW condition, when they are exploring and having a new experience. After that, if you allow them to go to sleep, you see that both the spines and the branches, these measures go down. And instead, very important, and that's the last column, the gray column, after the enrichment and the growth, if you keep the flies still awake, but in an isolated environment, so try to limit the learning as much as possible, you see they stay high. This is very important for us because, like the studies with the naps that I mentioned to you before, it means that once you have learning, it's not simply the passage of time that eventually will produce this controlled and renormalization. You really need the sleep to do this. Here is the evidence in mammals, in mice and rats. On the left, that's our original study in rats. In mammals, it's easier. There is one established marker, which is the expression of the AMPA, so glutamatergic AMPA receptors, the total number of the GLUE1 containing receptors, so their phosphorylation levels, increases with synaptic strengthening and decreases with synaptic weakening. There are hundreds of studies showing that, and that's why we use them, because they are very well established. And what you see is that we took very large brain regions, so either an entire half of the cerebral cortex or half of the hippocampal formation. Then we extracted the synaptic proteins and then measured them. And you see that overall, these markers tend to be 20, 30, 40% in higher expression after wake than after sleep. Again, this was taking animals after 6-7 hours of wake, spontaneous wake or short deprivation, relative to 6-7 hours of sleep. The study, again, talking about replication on the right, is by Rick Uganir's lab at Johns Hopkins, They basically then recently confirmed this finding in mice using this very even more refined preparation, which is this postsynaptic densities. They took the entire actually mouse forebrain, so almost the entire, brain of the mouse. And for them, this 100% line is the amount in wake. And relative to that, you see that many of these markers go down by 20–30% after sleep. So I think this molecular evidence is quite strong, and very importantly, you see this difference when you enrich for synaptic proteins, because that's where you should look, because that's where it counts that these receptors are expressed or not expressed after sleep. Again, it's very important that this seems to be true in very large parts of the brain. If this difference were to apply only to a very small specific circuit in the entire brain, well, maybe it would be interesting, but really probably hard to believe that that's the function of very fundamental function of sleep. Now, recently, we thought that another way, perhaps maybe the most stringent way to test these hypotheses was actually by using serial electron microscopy, basically measuring the size of synapses. And you need electron microscopy because you need to be very precise. And of course, the synapses are very small. So we have been using this block phase scanning electron microscopy that we have in the Department of Psychiatry. It's a wonderful machine that is able automatically to cut very thin sections of your block of tissue. Here, for instance, a piece of the mouse cortex. Automatically, you get this stack of images, maybe overnight. And then you reconstruct what you're interested in, that are the dendrites, the spine, the synapses, the axons. The problem for us has been, and still is, we have been using this for 6–7 years, is that the acquisition, the first part is relatively easy, but then the segmentation still is done manually. So it takes literally years to finish one study, because we need to reconstruct. Those are the pieces of dendrites, for instance, that we reconstruct. We try to reconstruct all what we see in a block. And then you see all the spines. Then the orange is the axon passing by. And then what we really care about is the red area that you see on the right. That's the axon-spine interface, which is literally the area of contact between the axonal button, the pre-synapse, and the spine head, the post-synapse. So it's basically that red line that you see here, but then, of course, in 3D is an area, because we know this is an established marker of synaptic strength, because fortunately for us, there is a very strong positive correlation between functional plasticity and structural plasticity. So if synapses get stronger, that's our hypothesis, they should also get bigger at the end of wake. And vice versa, if we are right, after sleep, they should literally, at least many of them, they should shrink. So that was our hypothesis. Again, in fact, this has been known in the structural plasticity literature. We could have chosen other markers. You see, for instance, the head, the volume of the spine head also is strongly correlated with synaptic strength, or the postsynaptic density in blue, or actually, if you really want to punish yourself, you can count the number of vesicles, which, by the way, one German student visiting wanted to do, and he did. They are all very strongly correlated, but we use the ASI, the red area, because for us it is the most reliable and quicker than other measures. But as I mentioned, it really doesn't matter, because all these, there is a lot of evidence, all these structural measures are correlated to each other. And most importantly, here you see that they are correlated to the functional measures. For instance, in the center, the synaptic area, which is what we are talking about, the red area, correlates with the total number of AMPA receptors. and the currents mediated by these receptors at the single synapse level. So that's why we use them. So every time we do this, it takes, as I mentioned, 4 or 5 people working full time for 3, 4, 5 years before we get to the results. And of course, we do this all blind. So the first study that we did was in the cortex of the mouse, primary motor and sensory cortex. In fact, the results that we found are exactly the same. So we pulled the data. So overall, there are more than almost 7,000 synapses. And then we did in the same animals, the hippocampal area in the CA1, and that's almost 8,000 synapses. In all these studies, as I mentioned also before, we always use these 3 groups. So we have a sleep animal that is sacrificed in the middle of the afternoon. These are mice, so again, they're nocturnal. So we monitor them, select them, and they have to be sleeping for most of the time, for 6-7 hours during the day. The spontaneously awake mouse is the one on top that is sacrificed in the middle of the night when we know they are awake, and we monitor them, of course. But we can't just use those 2 because they are differing in behavioral state, sleep and wake, but they are also differing in a crucial factor that is when the brain was taken, 3 a.m. versus 3 p.m. And we know there are many, many functions in the brain, gene expression, many, many things that change because of time of day. So we need to be sure that what we see is really driven by sleep and wake, behavioral state, and not circadian time. So that's why we have the middle group that is extended wake or sleep deprivation. So these mice are killed at the same time as the sleeping mice, but they are kept awake because literally we play with them. We give them novel objects for a few hours, and in mice it works very well. Of course, talking about stress, etc., these behavioral studies are actually, which is the beginning of our study, are difficult, and you need to be very careful in looking at the behavior. Even just giving a mouse a novel object can be very stressful. So we habituate the animals, we give them one new novel object every day. We habituate them to our presence for weeks, and then we do this experiment. When I tell you sleep versus wake, therefore, I always mean the sleep group relative to both wake groups, so that we can say probably what we see is due to sleep and wake and not to stress due to sleep deprivation or circadian time. So this is the main result that we found in this. The first study that we published was in this mouse cortex, in the superficial layers. Now, in this graph, each dot that you see is indeed one of these areas. So it's the area, the axon-spine interface in the sleep and in the 2 wake conditions. And you see the 2 wake conditions are really indistinguishable from each other. And you see that overall, the distribution of the ASI size in sleep is lower, smaller than in the wake conditions. On the right, you see exactly the same data. They're just plotted on a logarithmic scale. And this is just to show that the overall distribution of the ASI size in sleep is shifted to the left, meaning, at least at the population level, this is literally as down scaling, meaning every synapse goes down by an amount that is proportional to the original size of the synapse. In this study, we found that overall, this downscaling was by 18%. So just a few hours, 6-7 hours of sleep shrinks synapses by 18%. Now, crucially, and I'll go back to this issue of the selection, this process is widespread, but is not global. It's not that every synapse undergoes this scaling. For instance, in this study, at the population level, you see that the right side of the graph, so the largest synapses don't change, the 3 groups that totally overlap. We speculated that these are the strongest synapses, the one that, you know, are already committed. These are young, but already relatively mature animals. They are already committed to some kind of memories. They not easily change between just sleep and wake. But the majority do. So here is another way on the right to plot the data. The synaptic strength, the size of synapses in cortex is log normally distributed. This was already known. Basically, we find that the majority of synapses, 80% of the synapses, undergo this going down with sleep by 18% on average. These are all the small and medium size. But the large ones, the one with ASI by 0.8 to 1.6 in that graph, those do not change. Now, I just want to mention, we did also one study in pups. This is as early as we can go and still be sure that behaviorally we can distinguish sleep from wake in a pup. As you see here, this is the end, P13, so it's the end of the second postnatal week. These mice, of course, are still with the dam. You see them, most of them still have eyes closed. There is no way that we could have three groups. These pups sleep all the time, literally. So every hour they spend at least half of the hour asleep. So we don't have spontaneous wake. We have just sleep, and sleep deprivation, although it was extremely difficult, of course, to keep them awake and engage them. We were able sort of to do it for 3-4 hours, but not longer than that. But as you see, when we do this analysis, we still see a difference in sleep. There is overall a decrease. In fact, the decrease is larger than in the more mature animals. Overall is 34%. And in fact, this is global downscaling. So in this case, we see that all synapses go down. And again, we think that, well, perhaps it's because this is very early. The connectivity is still very much being built. And so every synapse is basically potentially subjected to this sleep-dependent down selection. Now, very briefly, in the same animals, we did this other area, which is the CA1 is one specific type of synapse, because of course you have to select which synapses to study. This is the CA3 to CA1 synapses in the middle of the stratum radiatum, one type, perhaps the most studied type of synapses in the learning and memory field. And is a little bit more complicated because the distribution of synaptic weight in this circuit is not log normal. There are two groups of synapses, the blue group, the middle and small synapses, and then there is a sizable actually portion of larger synapses. Take-home message, overall both types go down in size with sleep versus extended wake. In fact, the bigger group even changes in terms of number. So the number of synapses goes down with sleep. Now, the last study that we did, that we just published a few months ago, is in the cerebellum, because the cerebellum has always been, of course, intriguing and also challenging, because it seems in general that plasticity in the cerebellum and many other rules that apply to the hippocampus, the cortex, don't apply, or they are the other way around in the cerebellum. And so we wanted to test SHI in the cerebellum, also because you may know that the largest number of cells in the brain, in a mammalian brain, is the granule cells of the cerebellum, by far the largest, and by far the largest number of excitatory synapses are in the cerebellum, and they are indeed these ones, the ones from the parallel fibers of the granules to the distal dendrites of the Purkinje cells. So that's why we wanted to study them, and we studied them in the vermis of the cerebellum. Again, these are the same animals that we have used for the cortex and the hippocampus for the data that I've shown you. And now we look at the cerebellum, we do this reconstruction, again, almost 7,000 of these synapses. And you see, indeed, the cerebellum is different. This was already known. These dendrites with the as in red, they are super spiny. The density of synapses is enormous relative to this is typical of the cortex or the hippocampus. You also see the distribution is almost unimodal, meaning they're more or less, all these synapses look alike and are the same size, middle, small size. There is not such a variability as in cortex. And indeed, the cerebellum is different. When we look at the ASI size, there is no change. But very surprisingly for us, we do see a change in the number, in the relative proportion of these spines that have a synapse. So I didn't mention, but this is true in the cortex, in the cerebellum, in the hippocampus. The great majority of these spines have a synapse, more than 90%. Here in the cerebellum is 93% on average. And these spines, you see, it can be like single spine. They are like club-like with club shape, and then they have the ASI on one side. They all more or less look the same. Some 15% or so are branched. So there is a neck, and then they split, and both have the synapses. Only 7% of the spines are the blue one or are missing. So it's either a single spine or one of the two sides of the branch spine doesn't have the synapse. Okay. What we found is that it is the proportion of spines carrying or not carrying a synapse that changes in the cerebellum. So in the center is a representative dendrite from sleeping animals. You see many more of the blue. Those are the spine without a synapse. In fact, the proportion of we call them naked spines, is 10% in the sleep group and only 5% in the wake groups, which, if you think about, is striking that only in a few hours of sleep and wake, these spines can either acquire or actually lose a synapse. And we also found that this change happens specifically in the branched. So it's not the single spines, but they are the branched. They can lose one or actually even two of the synapses. And this, by the way, and I'll be happy to discuss this, may have strong functional consequences because the branch spines, because they have two synapses, are supposed to be much stronger and functionally with more, effect on the soma of the Purkinje cells. So the general rule that we see, which is very interesting for us, is that, and again, these are the same animals. The net effect of sleep is overall a decrease in the efficacy of the synapses. But you get there through different mechanisms, because in the cortex is all a change in the ASI size. In the hippocampus is both the size and the number. And in the cerebellum, it seems that it's really just the number of synapses that is changing. And this is just in the same animal. Of course, we think that, and we have some evidence I've shown you in the flies, that depending on the region, but also depending on the animal species, there must be different mechanisms to produce this down selection. Now, last part I want to tell you, so I hope I convinced you that we have converging evidence about a net change in synaptic strength between sleep and wake. What would be the benefit for the brain? Well, at the cellular level, it's kind of easy to imagine that I told you, synaptic activity is so expensive that just the fact that with sleep, many synapses get weaker is good, because then the brain can save energy and come back after sleep leaner and ready to go. is also this issue that, you can't keep going up because eventually you will saturate synapses. So bringing down many synapses prompts the brain for new learning. And indeed, there are quite several studies showing that after sleep, we learn better. Now, it sounds trivial, but I told you, if you are sleep deprived, every aspect of cognition is affected. So these studies very carefully controlled, for instance, for attention, because you could say, oh, sure, I don't remember because actually my eyes were closed. That's not what these studies are showing. They are correcting for attention, and yet they show a decreased ability to encode new information for learning. So that's perhaps easy to understand. What has been more difficult to understand is how can it be, probably many of you that, there is a lot of evidence that sleep is important for memory consolidation. It's still a very complicated topic. There seems to be some tasks that are not benefiting from sleep or not reliably, and we don't really understand why. But in general, there is strong evidence that sleep is important for the consolidation both of declarative memories and non-declarative memories. Many people then have a hard time to think, how can it be that, if during sleep synapses overall go down in efficacy, how does this translate to memory consolidation? So one idea that is, the active system consolidation by Jan Born in Germany and colleagues is that, well, no, actually during sleep, maybe there is downregulation, but there is also active net strengthening of some circuits that is happening. Now, we have been trying with Jan and many other people to really test this over the years. It's extremely difficult because you have to, bridge the gap between behavior, you have to find the right task, but then you have to be able to look at the synapses and follow the synapses before learning, after learning. It's very challenging. So recently, we have been able to do that for one task that we optimized in the lab, that is this complex wheel task, in which the mouse is put actually on the outer part of the wheel, and the wheel has an uneven rung pattern. So the mouse has to learn basically the sequence of these rungs. And of course, the wheel first is immobile and then starts moving faster and faster, and eventually the mouse will fall. But we measure its ability to stay up longer and not longer, basically. And so here is, you see a classical progression of learning. In 1 hour, the mouse learns to manage to, go to cope with a faster and faster speed. Now in the experiment then, and we have done this now 3 times, so we are very confident about these results. After the learning, session 1, some animals, mice, are allowed to sleep for 6 hours, others are sleep deprived for the same period of time. Then we bring them back to the wheel and we measure their performance. And what you see is that the sleep group starts at the same high level that was at the end of the session 1, while the sleep deprivation group starts lower. So we can say the sleep helps in the consolidation of performance in this task. Now, in the recent experiment, what we did was to combine this behavioral task, sleep-wake recordings, and in vivo 2-photo imaging in the primary motor cortex. Because we know, not because of our work, but other people's work, that these motor tasks are requiring plasticity in the superficial layers of the primary motor cortex. And so we apply this 2-photon imaging in which literally, without opening the scalp, just thinning the scalp, we can look at the dendrites and the synapses in primary motor cortex. These are genetically modified animals. That's why you can see the synapses. And actually what we look is this green signal, which is the expression of these GLUE1 AMPA receptors. And actually, in this particular case, the SEP marker allows you to be sure that these are receptors that are expressed on the surface. So they are really the receptors that count. And so the protocol was, first we look at this before sleep. After sleep, baseline, nothing happens. Then there is training, after training, and then post-training, other sleep or sleep deprivation. It's a very difficult experiment to do, but we managed eventually to collect 12 mice, an overall 1,500 of these synapses, again in primary motor cortex. What you show here, what you see here is the overall changes at the population level in these synapses in the conditions. So we see that after the first sleep period, baseline, overall the normalized difference, so the overall expression levels goes down, which is very consistent with what I've shown you until now. Then it goes up at time 0, which means immediately after the training. Not surprising again, this is what we know in the literature. This is expected. Then for us, the most interesting was, of course, the time 7-hour time point. If the animal is allowed to sleep, even immediately after learning, even if we just focus like here, specifically in the motor circuit that we know is involved in learning, the net effect of sleep is a going down of synaptic strength. And instead, if you sleep deprive the animals, the levels of the expression stays high. So this is important because one criticism that we often receive is that, well, everything that I've shown you until now was, you know, you give a novel object to an animal. Is that really learning? Which, of course, I just told you, we believe there is ongoing incidental learning. But in any case, this was an long-standing criticism, and I think this addresses the criticism because this is very specific motor learning, and we are focusing on that circuit. But of course, what we really wanted to do here was to focus on what we call the max synapses or learned synapses. So those are the ones that by definition increase in strength the most after training, which we find, so by definition we select them, we find that they are around 10%, again, very consistent with what is shown in the literature. So what happens to the max synapses at time, once we define, those are the ones that go up a lot at time 0. What happens afterwards? You see here, very surprising, interesting for us, actually, sleep or sleep deprivation doesn't matter. Once these synapses get strengthened by learning, even after 24 hours, they kind of stay very high, but the changes are exactly the same whether or not you have allowed immediately after training to sleep or not. The difference is not in the max synapses. The difference is in the majority, in the other synapses, the 90%, that were not directly involved in the training. Those are the ones that go down from training time 0 to 7 hours with sleep and instead stay high with sleep deprivation. So now, if you express the strength of the max synapses or learned synapses, not in absolute terms, but in relative terms, relative to everybody else in the circuit in that animal, and then you see that the relative strength in the sleep group of the max is higher than after sleep deprivation. So we think with this, and we also found, by the way, that the amount, how much the other synapses go down with sleep in their strength correlates the next day with how better the performance was in these mice. So overall, at least for this task, we can say that actually you don't need to invoke an absolute strengthening of synapses in a circuit during learning to account for the memory consolidation that comes after sleep. In this particular case, what we see is that the max spines stay at the same level, but the signal to noise ratio increases because it's the noise that goes down. And so together, you know, you get all the benefits. You go down in most synapses, so you have decreased energy needs, you save energy, you bring down, move away the circuit from saturation, and yet you also have memory consolidation. Now we are collaborating directly with Jan-Born to try to find another task in which we can test whether this is a general rule or not. Again, it's very difficult because you need a task in which you know exactly which synapses to look at, better be synapses that are on the surface that you can image. So it's very, very challenging. And then finally, that's my last slide. I promise to tell you that, you know, it's not a miracle on how we can get to this maintaining some synapses. The general take-home message is the following. First of all, we know that the fact, as I mentioned before, that neurons are active during sleep, it's very important. These two studies on the top right corner there show that, and these are not our studies, if you block the classical slow waves and the up and down firing of neurons during sleep, synaptic renormalization is abolished, which means that really you need to this firing to sample the synapses to do the down selection. The other study, the Japanese Norimoto study, showed that even in the hippocampus, if you block the sharp wave ripples that are basically the slow waves in the hippocampus, also you block the renormalization. So activity is very important for the sampling. And then here you see a list of some of the molecular markers that we think are important for this tagging. Eventually, when we talk about renormalization of synaptic strength in excitatory synapses, it means that at the end that you have to do the endocytosis of these glutamatergic receptors. And we know some of these markers, HOMER1A and GSK1 beta, they are important and they can be differentially affected by sleep and wake. I give you just one example and then I'm sure we can discuss later. GSK3 beta is always active, is in the spine. When it's active, it helps the constitutive recycling of these AMPA receptors. But when it gets phosphorylated, and this happens during learning, then the activity slows down. So you can see a mechanism in which you tag, because those were the synapses that became activated and engaged in learning, those synapses for several hours then are in a position, even if the animal goes to sleep, to actually be relatively protected by, from the down selection, simply because a core mechanism for this endocytosis has been impaired. And others like ARC are probably also important from that point of view. But again, I think this will be very good for the discussion. Thank you very much.
Thank you, Chiara. Such a great way to begin. For those of us who don't work at the level of power and elegance of what you've done. And thank you, Amber. And I'd like to now introduce Ryan Hampton, who's going to lead our discussion. And Ryan is a postdoc in the emotion training program. He's a social psychologist by training, so this was a big stretch for him, but he will do, I think, an admirable job of introducing our discussion.
Good morning, everyone. I just want to first say, in reading about your research and of course in this presentation, this is a really incredible set of findings, and the program of research that you have here to tackle this in different ways is really incredible.
Thank you.
So my first question basically brings us back to where we are today. We're at an emotion symposium. So in this research, you talk a lot about the impacts of sleep deprivation and sleep on cognitive functioning and on memory and things like this. But I wanted to ask, under this model, would you have specific predictions about how this kind of synaptic renormalization or lack thereof would influence the experience of emotions or the regulation of emotions, especially as it pertains with different phases of sleep like REM versus non-REM?
Yeah. In general, the expectation, the claim of this hypothesis would be that everything that is plastic, that changes for whatever reason during the wake period, has to undergo this process of renormalization. And so from that point of view, I have actually a hard time to believe that there is a single brain region that doesn't qualify for plasticity in wake. Because of that, definitely the circuits that are involved in the way we react to emotions or stress should follow the same rule. The challenge is to find the right way, the right circuit in which we can probe, as I mentioned, this question. There is quite sizable, not extensive, but sizable literature at the behavioral level in humans that I'm sure you are aware of about the general effects of sleep or sleep deprivation on mood in general, and then reactivity to emotions. You may have read the most, there were some interesting, intriguing, striking results. There are two recent meta-analyses of this literature, basically concluding that there is a lot of work to do to really make sure that sleep has an effect on emotions, and if it does, in which direction. It's very complicated, unfortunately, because of the design and because of the small number and because everybody uses a different paradigm in humans. Having said that, there is some evidence that at least the issue of intrusion, negative memories intrusions after a stressful effect, that at least in 2 or 3 studies consistently is shown to be positively affected, meaning the intrusion go down if the subject is allowed to sleep relative to a condition of sleep deprivation, which you can interpret as it could be that in some specific circuits that get strengthened during wake, then you allow this process to happen. But we have no, that I'm aware of, no evidence from animal studies in which we can study these circuits to support or not this hypothesis. There is, I didn't mention, so all what I've shown you until now is basically based on total sleep, which is made in a mammal of, 80% is non-REM sleep, 20% is REM sleep. Now, for the response to emotions and stressful situation, PTSD, etc., it seems the REM is the one that is supposed to be the most important because of the link with noradrenaline. And because REM is really the condition in which we know that's absolutely established, even more so now than 20 years ago, there is zero activity of the locus during REM sleep. So the expectation is that perhaps, but it's also true that non-REM has lower levels and non-REM is much longer than REM sleep. So overall, my inclination has always been to say that, well, this job overall of renormalization depends on both states. It depends probably also ontogenetically, at the beginning, REM comes before non-REM. Early sleep, active sleep in babies is mainly, REM sleep, because REM sleep is a brainstem driven. And so it might very well be that this condition of low levels of noradrenaline is good enough to produce the renormalization. It becomes more refined when there is no REM sleep, which can allow also circuits to be more locally regulated at the cortical level through the slow waves. But basically, one position could be that, as long as the noradrenergic levels is low, renormalization can happen, and they are both contributing. But unfortunately, again, I've shown you 3 regions. Now we are doing the striatum. It will take 2-3 more years. It would be fantastic to look in a model in the mouse at some circuits between, let's say, prefrontal and amygdala. But we need something that is very well established in which we can then know which kind of synapses specifically to look at, to test this.
Thank you. That's a very interesting perspective on that. I really, the aspect that you talked about relating to stress and trauma and how the normalization of the synapses during sleep after those events is such an important aspect of that. It's very interesting. Next, I just wanted to kind of broadly, this is a very technical, very scientific talk, broadly ask, what your thoughts about the implications of this model for balancing and maintaining a healthy sleep schedule is for people, especially as since a lot of psychopathology is treated partially simply by maintaining a regular and healthy sleep schedule.
Yeah, I totally endorse that. I am very protective of my sleep. All my students know that. I like to cook. I like to have people come out for dinner. But then when it's 9 o'clock, I say, you can stay here. I'm going to bed. I think sleep is very important. I definitely. Now, one issue is, though, I don't believe in strict rules. Everybody then asks, well, then how many hours do I need to sleep? And that is a dangerous and a difficult question to address. So I can tell you overall, the distribution is more or less a normal distribution. The majority of us need probably 7 hours or something like that. It changes with age. But the reality is that there are people who are perfectly functioning with much less than 7 hours or more than 7 hours. You are the judge. Only you can say how many hours you do need. And so, yes, I'm protective of my sleep, but I also tell people, you have to decide how many hours are good for you. This is true in humans, but now we know we used to, present the data, for instance, for sleep duration altogether at the, as a mean average. We don't do that anymore, even when we present fly data, mouse data. There is huge inter-individual variability, even when you study in this very controlled condition, in a lab condition, but there is very strong intra-individual consistency. Everybody has more or less a fixed amount of sleep that is needed. Why? Well, based on SHI, it would be that, some people are luckier than others, that the process of renormalization is more efficient. And why is that? It turns out the people who need little sleep usually have only the best part of sleep, the deeper part of sleep. They skip some of the early stages of sleep that make up almost half of our sleep. They go, they go immediately to the deepest stages where we think this process through the slow waves and then REM sleep can happen more efficiently.
So what about the role of like consistency in timing? So part of your research showed that these kind of patterns in pruning aren't explainable just by circadian rhythms. But is there a level of influence by the circadian rhythm to maybe efficiency of, like you said, how much time you're spending in deep sleep that may?
Yeah, there is definitely that. Now, we don't have, so I've shown you this evidence that it seems that the wake effect doesn't change in this very controlled condition between few hours of novel object exploration versus spontaneous wake. But our sleep condition is at the right time of day, which is the day for a mouse or the night for a fly. Now, there are studies in humans of especially with jet lag or shift workers, that many of the restorative effects of sleep, especially in the long term for the periphery, for the cardiovascular system, for instance, are just not there if these people constantly, chronically sleep at the wrong time of day. We have never studied in our conditions, if we can enforce a mouse to sleep 7 hours during the night rather than the day, whether we get the same effect at the level of renormalization. My guess would be that we don't, but it's not that, it's because simply the animal cannot sleep properly at the wrong time of day, which is also what happens in humans. So yes, they go to sleep, but they rarely get the quality of sleep that you can get during the, at night. Because, I didn't mention, but the circadian and the homeostatic system very much interact one with the other. And so if you miss one, the homeostatic will push you to go to sleep because the pressure is very high, but that doesn't mean that the quality is going to be always the same.
Great. Thanks so much for your responses.
Maybe we can open it up
yeah.
now for some questions. Let me, if I can, take the first question, Chiara. You mentioned in your comments to Ryan individual variability in sleep length, and you also indicated that it was found across species and that within a single organism, it's quite consistent.
Yes.
And so I wonder if there's any, I'm sure you have the data, whether the magnitude of synaptic reorganization is comparable in the long and short sleepers.
We don't have the data actually. So we would like to right now in our conditions, we try, to, as a first pass, we really select the animals. So only the animals that have slept 80% of the time for those 7 hours, those are our sleep group. So we actually select against this inter-individual variability,
yeah.
but definitely that would be in the long run what we need to do.
Yeah, because I wonder if in at least some variance in individual differences in overall sleep length may be related to the need for sleep-dependent synaptic reorganization, and whether among short sleepers there might be other mechanisms of synaptic reorganization that conceivably can occur during certain waking states that may supplement the synaptic reorganization that occurs during sleep. So I wonder if that's...
Yeah, that's a very interesting, it will be, it's possible. So my bet would be that, to do this process of renormalization, first of all, overall, it's a slow process because it's not just phosphorylation. You need to, let's say, to be specific about these glutamatergic receptors, excitatory synapses. So this is presumably something that requires many minutes, if not hours. So whatever you are thinking about is, something that you have to engage for quite a long time. Second, I stress this point about the fundamental difference between sleep and wake that allows the switching bias is the low levels of neuromodulatory systems in general. If you can have a state that you can maintain in wake, in which you are literally disconnected and with low levels of neuromodulators, then presumably you could do the job.
That's interesting. Yeah. And just a personal anecdote, you probably don't remember this, but many years ago, maybe 15 years ago, in a conversation you and I had, when we were talking about how the importance of sleep, and you were describing to me your story, that was when you suggested I give up using an alarm clock. And I did on that day, maybe 15 years ago, and have never used it since.
Oh, I'm glad to hear that. Yeah. That's another thing that I tell, my students, that's the best way to find out whether you are,
and my
sleeping
average
enough.
sleep increased by about 20 to 30 minutes.
Very interesting. Thank you. Yeah.
Okay, let's
But
open it
friendlier
up.
now too.
Hi.
There's one here and then that one, so we can.
Yeah, thank you very much. It was a really interesting talk this morning.
Thank you.
So as you know, I do mostly work with functional MRI, in particular resting state functional MRI. And throughout your talk, I kept trying to think of what implications this would have on our measures of quote unquote resting functional MRI, and in particular potential differences that you might observe if somebody is, say, drowsy or falling asleep in a scanner versus awake in a scanner. And in particular, if our interest is looking at individual differences in, say, trait-like characteristics or between, people who have depression versus not, how might those differ in a sleep state versus in an awake state? Because on the one hand, you showed examples that the synapses, or effectively connections, are changing. On the other hand, it's the same neurons, that also show very strong activity at awake, are also showing this change during rest or during sleep. So I'm curious what your thoughts are in terms of the ability of neural fluctuations during awake versus sleep to map out individual differences in, say, related to trait-like characteristics.
I think you need to be sure that you maintain. It doesn't matter when you calculate your measure of the resting state, you need to be sure that you can control or know about the behavioral state of the subject. So you probably know that, recently there has been publication showing that, in fact, from the MRI signal, you can, in fact, there are bold signatures of sleep and wake that are quite obvious, and that have shown that, in fact, what people, assume that when you go in the scanner, people are awake. In fact, very often they are not. And they are in a state of not REM sleep or not deep stage 3, but definitely stage 2, which is half of our sleep. So as long as you correct for that, and when you compare subjects, you know that they are in the same state, is probably okay. But that's assumption that they are awake to begin with. I don't think you can right now, based on that evidence, you cannot take that assumption.
Chiara, we have a virtual question here from our virtual audience. It is there a relationship of dreaming to memory consolidation and quote unquote smart selection of synaptic strengthening and weakening?
The first part of the question, so there is a meta-analysis study just published yesterday showing that indeed, if you do a task, there are 3 studies, so this is replicated. If you do a task like Tetris or some task, and then during during sleep, you dream about that task, after sleep, you are more likely to perform better in the task. The interesting twist is actually that this correlation was found for non-REM sleep, not for REM sleep. Perhaps simply because now we know, and this is a study that was done here actually in our lab by Giulio Tononi and collaborators, most of the dreaming happens in non-REM sleep, not in REM sleep. But so there seems to be actually direct effect of dreaming on the consolidation of these tasks that have been learned. How this is happening and whether this relates to, you know, bringing down everything that is not related to the task, so to the renormalization. Of course, these are studies done in humans. So the synaptic part, we have no idea.
Oh, I have the mic. Yeah, well, thank you so much. That was fascinating. And I had a few questions that I had to write down because I haven't slept in the past few days and I could not rely on my working memory, but I'll focus on two. So one question was about biological sex differences and if this, you know, whole process also differs across females and males. And, you know, I'm thinking about times as a new mom and long periods of sleep deprivation. And I hope I function to some extent, but, you know, so specifically, I was wondering about the hormones and how hormones interact with the cycle and affect this process. And the second part is about the quality of the sleep. So you showed a lot of data about the total sleep time, but I wonder if there's any differences in terms of people who wake a lot during the night and how this may affect the renormalization.
Yeah, so as I say, so this goes back to Richie's question. We don't really know about the variability. I mean, the assumption would be that what counts is the deep good sleep. So if we could select an animal that has only the stages, you know, N3 and REM, and compare to an animal that has only N2, the problem is that, you know, it's very difficult in mice to do that. But that would be the prediction. There is some differences in the overall sleep between, you know, depending on sex, but honestly, are minor differences. So they are not fundamental differences. In our data, we do these experiments in male and female mice, and the effects is the same. So we don't see major sex differences in these, you know, measures.
I have a couple of questions related to, I guess, sort of the neurochemistry at two levels. One is, can you say more about the mechanism by which the reduction in noradrenergic tone links up to the types of phenomena that you're talking about in relation to the molecular events and also the structural events? And then the second part is, you know, about medications that we frequently use in the treatment of patients related to sleep or related to depression or anxiety, or even psychosis. And do we know anything about the effects of those medicines on some of the processes that you're talking about?
Yeah, that part is easy. We don't. I mean, at least we have never done studies in giving mice some of these drugs and to see how they interfere. For the effects of the noradrenergic system, so in general, you know, we think I mean, there are several studies showing that in the brain, in vitro, in vivo studies, if you have no noradrenaline, it's easier to induce LTD, long-term depression, than long-term potentiation. At the mechanistic precise level, the study by Rico Uganir's lab that I mentioned, that study was able to link specifically the noradrenergic system to then one molecule, this immediately gene HOMER1A, and then the eventually down selection. So their story is the following. When we are awake, noradrenaline is high. HOMER1A is an immediately gene like FOS, gets induced because of neuronal activity in most of the brain or most neurons. We knew this, we have been known this for 20 years. But what they show is that it gets expressed and starts accumulating in the dendrites during wake. But then it can only enter the spines to reach the synapse when noradrenaline levels go down and adenosine levels are very high, which is exactly what happens at the end of a long period of wake. When you are tired, you are starting to go to sleep. Adenosine is very high simply because it's a marker of neuronal activity, basically. It comes from the metabolism of ATP. So adenosine is high, which signals that you have been awake for a long time. Noradrenaline has been high for a long time, but then drops. HOMER1A enters into the spines. HOMER1A eventually, through a very complicated mechanism, detaches the metabotropic glutamatergic receptors from the postsynaptic density. And eventually, because it doesn't interact directly with the AMPA, but eventually that then leads to the endocytosis of AMPA receptors. So that's a very elegant method in, pathway in which you can imagine, usually that's what happens in all synapses. HOMER goes in, and then suddenly all synapses are possibly in a condition to be depressed. So if this is true, then there must be other protective mechanisms for the synapses that we want to maintain because they've been, they learn the synapses. I mentioned one that can be this GSK. So as a positive protective mechanism, there must be others, but that's one possibility. But the precise link, noradrenaline and down selection that I am aware of is through HOMER1A.
Thank you. Other questions?
Hi, I'm Nasha. Thank you so much for that really intriguing presentation on synaptic homeostasis and the impact of sleep on it. I do research at the Sadhguru Center for a Conscious Planet at Harvard Medical Teaching School, and one of our studies is looking at the impact of meditation on sleep and on sleep deprivation. And I was really intrigued by this concept of looking at synaptic homeostasis. And I know that you mentioned that we don't yet know how to study it in humans, but I was just curious to know what do you think would be the impact of meditation on synaptic homeostasis?
Well, as I hinted to Richie, if you can meditate for quite a long time, keeping your neuromodulatory system very low, I would not rule out that you can do some kind of renormalization while awake. But yeah, I mean, I'm thinking about how to test this. The only way in humans would be to show some kind of, evoke response like the one that I've shown at the beginning, in which non-invasively, before and after meditation, you can show how effective the cortical connections are based on TMS. But I warned you before that, if you see a difference, if you see that indeed the slope goes down, is consistent, but is definitely not a very direct evidence, because just the balance excitation inhibition could change and explain the same result. So what you could do in humans to test this, I'm not sure. If you could do it, if you have the dream experiment in patients who are already implanted for other reasons and have deep electrodes in which you can stimulate, perhaps, and that would be a more direct, but of course this is, it's just not feasible. And on the other hand, I also joke, it's very difficult. The very important studies are the ones in humans in which you compare a nap with a period of quiet wake to make the point that you really need sleep. It's not just quiet wake, eyes closed. It's impossible to do the same experiment in a mouse. Mice do not have basically quiet wake. And so it's a fundamental challenge for using rodents, because the quiet wake condition is critical to understand the direct effect of sleep. And again, what I mentioned at the beginning, many people have many ideas about what sleep is for. But the bar for me is to show that that function cannot happen in quiet wake. And quiet wake, I think, is a feature of humans more, maybe primates, but not mice or flies.
Thank you so much. Yeah, hi.
Yeah,
Okay.
just one quick comment about this. I mean, that's a great way to frame it. And just to say that what we would say is that quiet wake is extremely heterogeneous. And so what we call quiet wake needs to be further refined, and the sort of course features may look similar, but looking at it in more detail, particularly
Absolutely.
biologically and phenomenologically, we can see a lot of variations.
Oh, absolutely. So, these studies which are already refined, the quiet wake means the eyes are closed, you try to not to get disturbed by the environment, you are not listening to music, you are not watching a video. That's quiet wake.
Yeah.
Okay. So that's the level of refinement. Yeah.
Okay.
Hi, really interesting talk. So you showed the P13 data and talked about how during that period, there's more of this global synaptic scaling that happens during sleep, but then in adulthood, it's much more refined. I was just curious if you've looked through any other periods of development, and is there kind of a linear transition, and how does the brain actually make those choices? And then does that correlate with how the need for sleep changes across sensitive periods of neuroplasticity?
No, we haven't. So it's P13, then most of the evidence that I've shown you is P28, P30, so it's late adolescent. And then some, for instance, the two photorepeated imaging is in adult mice. So it would be so interesting to be able to go even earlier, because P13 is still a lot of maturation, but is nothing relative to what happens during the first week. But there is no way for us to define really behaviorally sleep. And so that's a fundamental issue.
I had a question. When I saw you earlier talking about the rat pups and the amount of time they spend sleeping, it kind of got me thinking, wondering, because as organisms develop, and it's also very true with humans, the time points in which during the development in which sleep is more prolonged, what was the phrase? Apologies, just checking my notes real quick. Looking at the renormalizing the synapses, do you think that there are periods in neurodevelopment in which that basically dampening down, sorry, I'm not doing a good job, dampening down sorry.
I think one question, one interesting thing that we try to address is, at the beginning, like in the first week, or even in synapses are still growing.
Yeah.
And so you can imagine that during that period where we cannot even define sleep, maybe you don't need the renormalization because the circuit is still building up. Then eventually it's overcrowded and based on the environment and exposure, then you need the pruning. And perhaps that's when sleep starts to be needed. But it's total speculation because we don't know how to study sleep as a behavior. There are studies done, not by us, by people who studied, for instance, the development of the cortex. And so they take pups at P0, P1, P2, they take it for 20 minutes, they anesthetize with cold anesthesia. They look at how the brain, the firing or not firing, and that's it. That's the level of analysis that you can do during the first week. to relate these to sleep behavior is very challenging, if not impossible. But presumably at that stage in which you don't have synapses, actually, in the first week until P5, there are only few thalamocortical synapses. Corticocortical synapses are not there. So you could say, well, at least for the cortex, because, ontogenetically, phylogenetically comes later, you don't need sleep. But what about the brainstem? That's why I mentioned, REM sleep comes from the brainstem. Maybe for the brainstem, you need sleep the first week, but then we have to look in the brainstem, not in the cortex.
Hey, thank you. I'm coming from a landscape architecture field, so I was still able to understand that. So thanks. I was wondering if the amount or quantity of novelty that you would experience in a day affects the renormalization. I don't know if you can give the mice more new toys without stressing them out. But
Thank you for the question. Actually, absolutely. We have, because at the end, this hypothesis is about the cost of wake, but the cost of wake is the cost of learning and plasticity. So the idea would be that the more you learn, even if you don't extend wake, but your wake is very intense, then you need the renormalization. So we have been doing this in mice, in rats, in humans. In humans, you enrich you ask them to watch videos or audio recordings, only audio, and then you look at the depths of sleep in the auditory cortex rather than video, and then visual cortex. You see that the depth of sleep that is tracked by these slow waves increases specifically in those areas. So the entire subject, of course, goes to sleep, but that area has more larger of these slow waves, which we think are crucial to do the job of sleep. And the same in rats.
Hi, I'm thinking about how in humans, a lot of us go through these periods and during adolescence where we're just chronically sleep deprived and then maybe hopefully grow into adults with better sleep patterns. So what I'm wondering is if chronic sleep deprivation during key developmental windows like adolescence might have a lasting impact on the ability of these mechanisms to perform homeostasis. So if we're chronically sleep deprived in adolescence, if that might actually affect later on when we're adults, if we can actually do the same thing at a synaptic level.
Yeah, thank you for the question. So I don't know if that will affect the ability to do the renormalization in adulthood. I doubt that, but definitely I would hypothesize that it will affect, during adolescence, still there is circuit forming. That is affected by sleep deprivation. There are not many studies, but in animal models, in the prairie vole, and also we have a study in mice. If you do chronic sleep restriction during 1 month, so in mid-adolescence, the circuit forming in some areas of the parietal cortex, because it's one of the latest to develop, is affected. And so definitely, I think it's a safe assumption to say that, if you have a chronic, now in these conditions, it's severe and chronic sleep restriction that you enforce during adolescence, and you see some behavioral and structural effects later on in adulthood. I don't think, then is the circuit that has changed. In that circuit, probably then, if you sleep properly, renormalization will happen, but the circuit has been modified.
Hello, thank you for your talk. I'm wondering, with your TMS kind of studies, I guess I know with transcranial magnetic stimulation treating depression, they measure the resting motor threshold to then do the treatment. And I know after like a night of sleep deprivation or something, they'll have to remeasure the resting motor threshold. Do you have an idea as to why we would expect that to change? Is that kind of that LTD like affects the sleep deprivation?
Yeah. There are several studies that indeed that show that. Yeah.
Cool. Great. That's it. Thank you.
Yeah. So that's why you have to be careful when it's so critical, you know, to use exactly the same efficacy. You better control for this issue before sleep, after sleep. Yep.
Other questions?
I have another question. So I use EEG methods primarily when I'm doing neuroscience work. And when you were talking close to the beginning about, you know, part of the necessity of this model, like you said, is that it has to be able to explain why we're going offline. And when you talked about that, talked about this kind of exhaustive sampling across the brain and about neurons firing largely in synchrony. And throughout sleep, you go through you know, these kind of synchronous waveforms at different frequencies. And I was wondering if you would hypothesize that potentially different groups of neurons or different parts of the brain or different types of renormalization are happening in these different frequency bands. And
the frequencies I was mainly referring to is the major frequency during non-REM sleep, which is the very low frequency, so 0.5 to 4, which is basically the slow waves. And so in the slow waves, neurons are either firing for half a second and they stop firing for the next, and then they keep. So that's the, it would be very difficult. I don't think, so why slow waves? Because we know in vitro, strong firing followed by silence and hyperpolarization, because we know from our animal studies, on-off firing is not just on-off, is depolarization followed by hyperpolarization. That is a perfect condition to produce depression. So that's why, you know, it's natural for us to think that, well, that's a mechanism, at least in mammals. I used to say, you know, in flies, there must be another one because there are no slow waves in flies. I was totally wrong. Now we know there is at the population level slow wave activity in the central complex of the fly. So it may even be true for flies, but that's for theta frequencies in REM sleep or specific, much more localized frequencies. I don't have any direct evidence to suggest that, you know, that frequency in sleep would be conducive to depression.
Thank you.
Thanks. I had a couple of questions. One was to do with sleep deprivation and pain. So there seems to be some evidence that after prolonged sleep deprivation, there is pain, and also how that might be linked to emotional reactivity.
There is some evidence, and in fact, we are trying to test the idea that in fact is, so there is lower threshold, so higher sensitivity for pain. And perhaps it's because some specific circuits have been strengthened by the being awake, basically. And so we are trying to find the right model, again, the right circuit and synapse in which we can test this directly.
Okay, so there's no direct evidence yet on what the mechanisms are?
Yeah, I don't think there is direct evidence.
Okay, thank you. And the other question that I wanted to ask was, in terms of those with dementia, they seem to have very good memories of their younger ages, but forget things very rapidly when they, you know, whatever they've learned just now. And would that sort of link with the normalization hypothesis that you were mentioning, that stronger synapses are sort of intact and the weaker ones are just normalized out? You say? Yeah,
probably. Well, you know, it's really speculation, but it would be some synapses, the ones that you have enforced and you have really locked to some memories early on, and those don't change. In the dementia, then the issue is to begin with is not sleep, although it's a vicious circle, is the wake condition in which you don't encode, you don't form strong synapses, and then there is even no need for renormalization, but it's both probably, but it's all speculation.
Hi, this young woman's question about TMS and MDD and sleep deprivation jogged my memory that we can use sleep deprivation to sort of temporarily treat symptoms of major depression. Is there any evidence that there's something to do with like renormalization or like something else that has that temporary effect on that?
One hypothesis was that is still, it's very difficult to test, is that in these people, there is no enough build-up of synaptic strength during wake. And that's what you do with sleep deprivation. And then because of that, if you build up the pressure, then sleep can happen in a consolidated way, and the process can be efficient. But if you don't have enough pressure because you have not been long enough awake, or in this case, because basically there is not enough learning and plasticity, that's then there is little pressure and need for sleep, and then it's a vicious circle.
I'll make it quick. Thank you for your presentation. I'm curious as to your thoughts about using fear conditioning and maybe extinction recall as a paradigm to gauge sleep's association with emotional learning.
Yeah, we are trying. Is one of the, the two kind of tasks that we are trying to use is one is this, although it's not the kind of task that usually people are associating with benefits of sleep, simply because I think this is one type of tasks that you have 1 second learning, you won't forget easily. Or put it in another way, you don't need the sleep or the sleep deprivation to see an effect because it's too strong of a learning. When I hinted to the fact that, there are forms of learning that do not benefit then in terms of consolidation from sleep. And in general, if you train super well and with wake, it's much more difficult to see the effects of sleep in terms of performance. Fear conditioning is such a strong learning that it is difficult. So I wouldn't know where to begin with because there is no nice behavioral paradigm in which I can say, sleep makes a difference. And therefore, then I can look at the circuit that can explain this either as further strengthening sleep or weakening.
Thank you, Chiara.
We'll resume at 10:45. Thank you.